Hello, welcome to Biology, the scientific study of life. Tests in humans. The receptor cells for tests in humans are modified epithelial cells organized into test buds, which are scattered in several areas of the tongue and mouth. Most test buds on the tongue are associated with nipple-shaped projections called papillae, in addition to the four familiar tests: perception, sweet, sour, salty, and bitter. Fifth, called umami, Japanese for delicious, elicited by the amino acid glutamate and perhaps others. Glutamate is the key component of the flavor enhancer monosodium glutamate MSG, and is abundant in foods such as meat and aged cheese. Each type of test receptor can be stimulated by a broad range of chemicals, but is most responsive to a particular type of substance. Complex flavors are perceived as the brain integrates the differential input from many test receptors. Transduction in test receptors occurs by several mechanisms. Chemoreceptors that detect saltiness, mainly the presence of sodium ions in A positive, and sourness, hydrogen ions H positive generated by acids, have channels in their plasma membrane through which these ions can diffuse. The influx of Na positive and H positive depolarizes the cell. The mechanism of umami chemoreceptors may involve the binding of glutamate to Na positive channels. When glutamate is bound, the channels open. Na positive diffuses into the cell, and the cell depolarizes in some chemoreceptors that detect bitter substances such as aquanin. The substance binds to potassium ion K positive channels and closes them. The resulting decrease in the membrane's permeability to K positive depolarizes the cell. Finally, sweetness is detected by chemoreceptors that have receptor proteins for sugars. Binding of a sugar molecule is to a receptor protein triggers a signal transduction pathway that results in depolarization. In all test receptors, the depolarization causes the cell to release neurotransmitter into a sensory neuron, which transmit action potential to the brain. Smell in humans. Olfactory receptor cells are neurons that line the upper portion of the nasal cavity and send impulses along their axon directly to the olfactory bulb of the brain. The respective ends of the cells contain cilia that extend into the layer of mucus coating the nasal cavity. When an odor substance or odorant diffuses into his or this region, it binds to specific proteins called odorant receptors, ORS, on the plasma membrane of the olfactory cilia. The binding triggers a signal transduction pathway involving the G protein, the enzyme adenylyl cyclase, and the second messenger cyclic AMP. The second messenger opens channels in the plasma membrane that are permeable to both Na positive and Ca positive to the power ions. The influx of these ions depolarizes the membrane, causing the receptor cell to generate action potentials. Human can distinguish thousands of different odors, each caused by a structurally distinct odorant. This level of sensory discrimination requires many different ORS. There are more than 1,000 OR genes, about 3% of all human genes. Each olfactory receptor cell expresses only one or at most a few OR genes. Cells with different odorant selectivities are interpreted in the nasal cavity, but their axons sort themselves out in the olfactory bulbs. The cells that express the same OR gene transmit action potentials to the same small region of the olfactory bulb in bulb. Although all receptors and brain pathways for taste and smell are independent, the two senses do interact. Indeed, much of what we call taste is really smell. If the olfactory system is blocked, as by a head cold, the perception of taste is sharply reduced. Locomotion requires energy to overcome friction and gravity. Um, Most animals, however, are mobile and spend a considerable portion of their time and energy actively searching for food as well as escaping from danger and looking for mates. Our focus here is locomotion or active travel from place to place. The modes of animal locomotion are diverse. Most animal phyla include species that swim on land and in the sediments. On the floor of the sea and lakes. Animals crawl, walk, run, or hop. Active flight, in contrast to gliding downward from a tree or elevated ground, has evolved in only a few animal groups. Insects, reptiles, including birds, and among the mammals, bats, a group of large flying reptiles, died out millions of years ago, leaving birds and bats as the only flying vertebrates. In all its modes, locomotion requires that animal expend energy to overcome two forces that tend to keep it stationary: friction and gravity. Exerting force requires energy-consuming cellular work. Swimming. Because most animals are reasonably buoyant in water, overcoming gravity is less of a problem for swimming animals than for species that move on land or through the air. On the other hand, water is a much denser than more viscous medium than air, and thus drag friction is a major problem for aquatic animals. A sleek, fusiform troubadour-like shape is a common adaptation for fast swimmers. Animals swim in diverse ways. For instance, many insects at four-legged vertebrates use their legs as oars to push against the water. Cichlids, scallops, and some cnidarians are jet-propelled, taking in water and squirting it out in bursts. Sharks and boneless fishes swim by moving their body and tail from side to side, while whales and other aquatic mammals move by undulating their body and tail up and down. Locomotion on land. In general, the problems of locomotion on land are the opposite of those in water. On land, a walking, running, hopping, or crawling animal must be able to support itself and move against gravity, but air poses relatively little resistance, at least at moderate speeds. When a land animal walks, runs or hops, its leg muscles expend energy both to propel it and to keep it from falling down. With each step, the animal's leg muscles must overcome inertia by accelerating a leg from a standing start. For moving on land, powerful muscles and strong skeletal support are more important than a streamlined shape. Diverse adaptations for traveling on land have evolved in various vertebrates. For example, kangaroos have large, powerful muscles in their hind legs, suitable for locomotion by hopping. When a kangaroo lands, tendons in its hind legs momentarily store energy. The farther the animal hops, the more energy the tendons store, analogous to the energy in a compressed spring on a pogo stick. The energy stored in the tendons is available for the next jump and reduces the total amount of energy the animal must expend to travel. The pogo stick analogy applies to many land animals. For instance, the legs of an insect, a dog, or human retain some energy during walking or running, although considerably less than those of a kangaroo. Maintaining balance is another prerequisite for walking, running, or hopping. A kangaroo's large tail helps balance its body during leaps and also forms a stable tripod, with its hind legs when the animal sits or moves slowly, illustrating that same principle. A walking cat, dog, or horse keeps 3 feet on the ground. Bipedal animals such as humans and bears keep part of at least 1 foot on the ground when walking. When an animal runs, all 4 feet or both feet for bipeds may be off the ground briefly, but at running speeds, it's momentum more than foot contact that keeps the body upright. Crawling poses a very different situation because much of its body is in contact with the ground. A crawling animal must exert considerable effort to overcome friction. You have read how earthworms crawl by peristalsis. Many snakes crawl by undulating their entire body from side to side, assisted by large movable scales on the underside. A snake's body pushes against the ground, driving the animal forward. Boa constructors and bathons creep straight forward, driven by muscles that lift belly scales off the ground, tilt the scales forward, and then push them backward against the ground. Flying. Gravity poses a major problem for flight because wings must develop enough lift to overcome gravity's downward force. The key to flight is wing shape. All types of wings, including those of airplanes, are airfoil structures whose shape alters air currents in a way that helps them stay aloft. Flying animals are relatively light, with body masses ranging from less than a gram for some insects to about 20 kilograms for the largest flying birds. Many flying animals have structural adaptations that reduce body mass. Birds, for example, have no urinary bladder or teeth. A fusiform shape helps reduce drag in air as it does in water. Comparing costs of locomotion. The study of locomotion returns us to the theme of animal bioenergetics. The energy cost of locomotion depends on the mode of locomotion and the environment. Running animals generally expend more energy per meter traveled than equivalently sized animals specialized for swimming, partly because running or walking requires energy to overcome gravity. Swimming is the most energy efficient mode of locomotion, assuming that an animal is specialized for swimming. And if we were to compare the energy consumption per minute rather than per meter, we would find that flying animals use more energy than swimming or running animals with the same body mass. The downward slope of each line on the graph in this figure also shows that a larger animal travels more efficiently than a smaller animal specialized for the same mode of transport. For example, a 450 kilogram horse expends less energy per kilogram of body mass than a 4 kilogram cat running the same distance. Of course, the total amount of energy expended in a locomotion is greater for the larger animal. An animal's use of energy to move determines how much energy in food it consumes is available for other activities such as growth and reproduction. Therefore, structural and behavioral adaptations that maximize the efficiency of locomotion increase an animal evolutionary fitness. Okay, thanks for listening.